Right, okay, so today's lecture goes under the name of A Tale of Two Databases. It's an basically I'm going to talk to you about some of the research and the work that I've done in the past and hopefully pitch it at least reasonably towards the database context that we've been thinking about all term. So there's a bit of databasing, bit of analysis, bit of data. It's an overview of some of the stuff that I've been doing and some of the problems that I've faced. Now, it was called A Tale last year. The talk was A Tale of Two Databases, and I talked about two databases, my Mexican database, and the second database that I talked about was one that I worked at in my previous job called the Entangled Bank. Now, I kind of ran out of time last year to talk about Mexico, and I wasn't very satisfied with how the talk went. So this year, what I've done is we're just going to have Mexico, part one, and we'll have part two next semester because the Entangled Bank is a web-enabled database and therefore sits better with the web development stuff that we'll do next time. So we're not going to not have it. It's just that I've got so much to talk to you about Mexico that I've filled the period with Mexico. And we'll do the Entangled Bank next year. We'll have part two next year. OK, so it's a tale of one database. So I hope you don't feel too short-changed yet. OK, a little prologue. So I suspect I am virtually the only person, or certainly 5-10 years ago when I started this, I was probably the only person in the world who had both a GIS qualification who was interested in evolutionary science. So to a certain extent, I want to put some of the research, I have to put the research that I'm going to talk to you about in context. I have to tell you, or I'm going to tell you, whether I have to or not is another matter, going to tell you about some of the history that led up to what I'm doing and why I'm doing it the way I'm doing it, perhaps. Anyway, it always helps to know where people and what have come from. So we'll start off with Charles Darwin and the voyage of the Beagle in 1831 to 36. Charles Darwin left, I can't remember whether he lived, I don't know which port he went, travelled around the world via the Canaries, down South America, famously to the Galapagos Islands, where he studied the Galapagos finches and the tortoises, all the way around the world through the Society Islands, round to New Zealand, Australia, and back to the United Kingdom. One of the great scientific voyages of discovery, well, not really of discovery, of biological discovery. They weren't discovering new lands at this point, but they were scientifically investigating. It was a scientific expedition. We must also not forget that on the other side, Darwin travelled the world. He's the most well-known scientist associated with the theory of evolution. However, the theory was also independently devised by Alfred Russel Wallace, who spent most of his time collecting plant and animal specimens in Indonesia and the Far East. And in particular, his most famous discovery was something called the Wallace line, this line drawn on this map, which is a major biological division between species. You have Australasian species on this side of the line, you have Asiatic species on the other side. And it was this traveling around the area and seeing what species lived in particular places that led Alfred towards the theory of evolution, just like it did Darwin. And these, of course, were Wallace and Darwin were the co-authors of the first paper on evolution published at the Royal Society. And I think you can see from this map, maybe not from the previous one, that geography was an integral part of the development of the theory of evolution. But of course, Darwin's name is most associated with the theory, published The Origin of Species in 1859. And in chapter 5, there is one of the classic quotes out of The Origin of Species. The other one, incidentally, relates to the Entangled Bank, the database I'll talk about next year. So my life has involved two of the greatest quotes from one of the greatest books ever written. So this quote is, "The affinities of all the beings of the same class, the same type, have sometimes been represented by a great tree, the tree of life, which fills with its dead and broken branches the crust of the earth and covers the surface with its ever-branching and beautiful ramifications." I guess one of my primary aims in research over the last 10, 15 years is how do we turn that vision into a reality? How do we map the branches of evolution, how they spread across the globe, how the history of life on Earth, basically? How do we do that? So the representation of life as evolutionary patterns as trees goes back a bit beyond Darwin. It goes, in fact, the first published diagram of a tree-like structure in relation to evolutionary studies comes from Lamarck. As you can see, he's sort of a generation previous. Well, he's slightly older than Darwin. But Lamarck is, perhaps you may have heard of Lamarck, but he's not. He didn't become the father of evolution because he didn't have a method. He could see the hierarchical structure and relationships between the organisms that he studied, but he didn't know how it came to be. He believed it was through our individual lives. We have struggles in our lives, and therefore that changes our bodies, and therefore we pass those changes on to our offspring. We know that's not, in fact, how it works. So Lamarck drew the first tree, but he didn't have a process. And Darwin and Wallace's key contributions to the study of evolution was to elucidate the processes of evolution. This is Darwin's first, as far as we know, Darwin's first drawing of a tree in relation to evolutionary patterns in his first notebook in 1837. And indeed, in the first edition of the Origin of Natural Selection, the only figure in the entire book is this tree figure, which is actually representing pedigrees, as in of things like dogs and fathers and mothers and children of dogs. But this is the only diagram, tree-based tree diagram in the Origin of Species. Since then, there's Darwin's diagram in 1859. This is a paper from Godfrey Hewitt, of University of East Anglia, where he traced how the development of iconography, drawings, graphics, the graphics that we use to represent and describe evolutionary patterns from Darwin's first tree up till, in this case, the beginning of the millennium. We can see these early trees by Darwin and Haeckel all right, we don't see the detail, but they're quite ornamental. They look literally like trees. They're drawn literally like trees, some of them. And then as we move on into the post-war period, we get a huge expansion in the different ways that people have drawn evolutionary trees, reflecting different aspects of the study of evolution, whether we're talking about the biogeography of islands and migration between oceanic islands. Here's the Canary Islands, movements between the various Canary Islands on the left-hand side, different ways in which that geographical movement has been shown. Here in the middle, we have trees, evolutionary trees, drawn across space with a spatial element. And then we've got a set of trees here with various attributes associated with them, traits of the organisms. And here, finally, on the right-hand side, we have what's called the coalescent, which is basically gene trees embedded within species trees, but we won't worry about that. Take-home message from that picture is that there's been an evolution through time of the information that we've got associated with these evolutionary trees. We've got more and more stuff that we want to annotate the tree with. And of course, from my interests, I'm particularly interested in these two geographical pathways. How do we display evolutionary trees into geography? This is just an example. This is actually getting old, a bit old now. This is probably at least 10 years, a decade old. This is, at the time, was the most detailed evolutionary tree of all organisms that we had. You are here. We are just part of this massive tree. This is time, by the way. We're looking at a circle here. These are the ancestors, and this is time in this case going out. So this is showing the kinds of trees that are being produced now, and there's probably trees that are 10 times this size now. So I've been waffling on about evolutionary trees, but I haven't actually told you what one is yet. I'm kind of expecting a reasonable knowledge from you, but here we go anyway. So what is an evolutionary tree? It's a hierarchical model of similarity or descent, and we're particularly interested in descent. So in this diagram, the human is more closely related to the mouse than we are to the lizard. We're the human, the lizard, and the mouse are more closely related to than the frog. Okay, that's how you read them. Importantly, we've got it drawn on a piece of paper here, but a tree doesn't exist in two-dimensional space. It exists in what we call tree space, the space of trees. So, for example, we can see here that this is in fact the same tree, but just drawn in different ways. So we've got these topological isomers. So here we've got the human and the mouse are most closely related, and they're most closely related to the lizard, but we've just drawn them differently. So the same tree can be drawn in different ways. Two types of evolution. There is what we call microevolution, which is evolution within a species. It's all to do with demographic, who mates with who, and genetic processes, who survives. So it's control. We're interested in, if we're studying microevolution, we're interested in mutation rates, the dispersal of individuals, natural selection on individuals, which members of the population survive, which ones die, which ones pass off on their genes to future generations. We're also interested in local population extinctions and things like that. And that in a microevolution, individuals, think of humans, interbreed to form a population network. So it's not a tree structure. When you get individuals all mating together, then you get a population network, a reticulate network of relationships between individuals. But when subnetworks, maybe this area out here, these red ones here, get isolated in some way from the rest of the population, then slowly mutations build up, natural selection operates on these mutations, and if there's no dispersal between these populations, it's cut down, that can lead to speciation, the formation of new species, two new species from one original species. So that's microevolution. Macroevolution is the pattern of evolution among species at large spatial and temporal scales. So it's the relatedness between groups of species that cannot interbreed. So we're interested in the processes of speciation, creation of new species, which is basically the microevolution part, but also dispersal and extinction of species across geographical space. How do we build these evolutionary trees? Well, we infer them or calculate them from measurements on living organisms and fossil organisms. So, of course, for a lot of the work that's done today, we're talking about DNA sequencing of different species and then building trees from those species. But with fossil organisms, we don't have many DNA or very little DNA. So it's done from things like measurements of bone sizes and all sorts of stuff like that. Importantly, for macroevolution, that always results in a hierarchical tree or phylogeny of descent. So phylogeny is the word used to describe an evolutionary tree. So if I say phylogeny, it just means evolutionary tree. Just a little bit about trees, roots, and times, just so that we understand what we're looking at to a certain degree when we see things in due course. There are various types of tree models. This is not exclusive to evolution. They could be trees that represent other things, don't have to be, but I'll talk in evolutionary context. So we have what we call unrooted trees, where it's just the relationship between these various groups is simply the distance along the network. But we don't know, there's no time involved here because we don't know there's no where things have evolved from. It's just similarity, these unrooted trees. But what evolutionary scientists do is they do something they call root their trees. Now, I'm not going to go into how they do it, but basically they say they have a way of identifying where the oldest point is on the tree, and then we orientate the tree from that route. Don't worry about how it's done. And we can get two types of trees. We get trees that sometimes are like this, with dying with endpoints. And these trees may, so if you go and do some DNA analysis on a whole load of taxa, and then you basically measure how different each species is from each other, and then you use that information to build the tree, okay? When you do that from DNA, you don't have any time. All you do initially is have how different are we from each other. So this kind of tree is showing simply how different they are, but we know where the start point in time was. So we can say that these two parts of the tree we know they're younger than this part of the tree, but we can't do any cross-branch comparison. So this part of the tree, this speciation event here, we can't say whether that's the same time. That may in fact be younger than this one. It's just that more differentiation has happened in the DNA since they split. We can also have rooted trees which are, we've calibrated to time. So we've basically stretched these branches. So we build these trees like this. Then we have some fossils or some kind of evidence that allows us to calibrate the tree, stretch it into time, turn genetic differentiation into time. Now, there are all sorts of issues about how you do this. Not going to go into it, but basically we might end up with trees like this and trees like this. And in this case, the distance along the branch is represented in time because we've calibrated it to time. In this case, this is a tree. All the organisms are living. So if we did a DNA analysis on various organisms, ended up with a time-calibrated tree, it would look like this because all the organisms were still alive. However, if we do it with fossil organisms, we might end up like this. So this might be the evolutionary tree of human species. We've only got one still alive today, Homo sapiens, but in the past there have been various Australopithecus and other species which have then died out at a particular time. These diagrams, particularly diagrams that are drawn from or phylogenies that are derived from modern data, of course, don't include the extinction. This was pointed out by Stephen J. Gould in the 1990s or 80s. So we may see a diagram like this. So we do modern DNA analysis on a whole load of existing species. It looks like we've got an increase in diversity through time. But because we're only sampling this part of the tree, all this stuff, hidden stuff in the past may be extinct. We can't see this from the modern organisms. We need the fossil organisms to be able to do this. So bear in mind that if you're doing modern studies, you may see a tree that always diversifies, but that doesn't actually mean that there are more species today than there were in the past. It's just an artifact of the fact that you can only sample living organisms. Finally, do you draw them upwards or downwards? There ain't no, basically, there's no real great reason, but or people draw them different ways. So tends to be, so family trees tend to be drawn from the past down the page towards the present. So when we're talking about shallow time, trees are quite often drawn this way because they're mirroring a family tree sort of thing. Deep time, geologists, so geneticists maybe draw trees that way. Geologists maybe draw trees this way, because I think they draw them this way is because they stand there an awful lot of time looking at cliff cliffs where the youngest sediments are at the top and the oldest ones are on the bottom. So it's understandable that you draw your tree orientated to the thing you're interested in. Now, generally, this supports the tree metaphor in that it looks like a tree. That would be more like a root metaphor, and it looks like a set of roots. But in my case, this is a more sensible way to draw it because you can sit trees like that in 3 dimensions on top of maps, which is what I'm going to do in due course. Okay, so geography, having gone through a crash course in tree reading and evolutionary science, phylogeography. So this this is not 2003, so we're talking 13 years ago when I started my work. This was the kind of most advanced diagram that you might get showing a tree in geography. These are snails along the Western Australian coast. Here you can see you've got a tree, evolutionary tree, and basically they've just, luckily it's a nice linear system with the tree fits well with the linear nature of the coastline, and you can just draw from these ranges. These are the ranges of the organisms at the tips, and they can be connected to the tips of the tree graphically. It's just a graphic image. So there we were in 2000. So that was the state in 2000. And 2003, I was probably, although my memory of the dates is so appalling I couldn't tell you to be honest, at the University of St Andrews with Mike Ritchie, my PhD supervisor in the days when I drank. We would sit in the pub and talk things over. And Mike's interest is in the sex lives of crickets. And I was working on bush crickets. This is called a Phippiger, saddleback bush cricket. Lives all across Europe. We spent a lot of time in the south of France in the Pyrenees catching them. Interesting, well, a few asides, it's Christmas. Here's a male, this is the male of the species. Mike was particularly interested in them because of this. This is called a spermatophore. So the male is basically a present, a nuptial gift between the male and the female. So the male produces one of these every about 2 to 3 days, which must cost him quite a lot of energy. He gives it to the female. The female will eat. Most of it's proteinaceous. She eats it, and then she decides whether she fertilizes herself with the sperm that's associated with it. Anyway, that's an aside story. We spend a lot of time thinking about insect sex. We spend a lot of time measuring their genitalia and other exciting things like that. While I'm in the Pyrenees, this is Constantino Marcias Garcia, who is now head of the Instituto de Biológica at UNAM, University National Automada in Mexico City. They are both interested in sex. In Mike's case, it's insect sex. He spends his entire life nowadays measuring the calls of fruit flies and how they talk to each other. Tino, who a friend of Mike's, used to come over to St Andrews on various visits. His passion is fish sex. So he had spent many years studying the sex lives of a group of fish in central Mexico, and indeed Mike was helping him out with that research on the genetic side of this research. And Tino was interested, and Mike are interested in some of these fish, is because they're sexually dimorphic. So here's the male, here's the female, and mating they were measuring basically, do males with bigger fins mate with more females? Do females like big fins, or do they not like big fins? All that kind of stuff. So the sex lives of fish. Clearly, I'm not hugely interested in the sex lives of fish, but I am interested in the geography of speciation. Of course, the sex lives of fish influence how they evolve, but so does their geography. So the question Tino came to me, as we sat around in southern France after a hard day's insect collecting, was how much of the speciation in the group of fish that we're interested in is due to their sexual behavior, and how much due to their geographical history. That's kind of the question that we wanted to answer. Now, while I was writing my PhD, one of the products of it was this paper called Phylogenetic Information Systems, putting the geography and phylogeography. It's a review type paper in the Journal of Geography, and it was saying sort of basically, wake up world, this is GIS, evolutionary scientists, you're not using this amazing system. Here's some of the things we might be able to do with it. And one of the things that I did, just as a demonstration piece, was this simple visualization, 3D visualization in ArcScene of various fish species in Mexico. One of them, I think the purple one being the one that Tino was interested in, some other groups of fish as well, looking at the histories. These are the river channels in the Miocene, the Pliocene, and the present day, and saying, look, we can visualize this information, we can explore it, and this allows us to explore it and explain the patterns to people, and it's probably a good idea. And of course, if we go back to the quote by Darwin, this is the first effort in trying to take that evolutionary tree and its dead and broken branches through the earth. Well, this is the first attempt to do it. And as far as I'm aware, it's the first ever GIS-based visualization of an evolutionary tree in space, as far as I know. And that paper, plus, just my general research interest, got me a postdoc. Once I finished my PhD, a place called the National Evolutionary Synthesis Center, which is in Durham, North Carolina, where I spent 3 years playing with this question: how do we visualize the history and analyze the history of life on Earth across the planet? And that's how I got to be doing this research on Mexico. So, Mexico is a very highly biodiverse country. It's one of the major biodiversity hotspots in the world. Why is it so diverse? One, it has a huge range of environments, from the tropics, tropical forests down in Cancun, up to high mountain plateaus, mountain species up here. We've got deserts, so it's a highly varied ecological environment. But also, it's its history, its geographic history. So it's the contact place between the near Arctic, which is species that evolved in North America and the North American continent, and species from the neotropical that evolved in South America. So roughly, they don't really know when, but between sort of 20, 25 million years ago, North and South America crunch together, meet, the Panama Isthmus forms, and we get the development of a huge mountain range in central Mexico. So we've got species from South America making their way north, for example, the armadillo. That's an example of a species that made its way north. And then we've got species, sorry, not the armadillo, the opossum. The American opossum the marsupial opossum came down up from South America. We've also got species coming from North America and making their way down south into. But we've also got this mountain range building, a huge mountain range, 14,000 feet high in roughly 15 million years. And this turmoil in here generated lots of what we call endemic species. Endemic species evolved here, they only live here, and they're only found here. So it's what we call a hotspot of endemism, plus we've got a mixing of two biota, and that's why it's such a highly diverse place. So, this is a study. I've been working on what we call the historical biogeography of the Goodeidae subfamily. The Goodeids is just the name of the group of fish that we're working on. And basically, the questions that we're kind of asking is, how did they get where they are now? What's their history? How did they come there? And what elements of the environment drove the diversification in the group? So we've got 32 to 35 species to central Mexico, endemic, as in they only live in central Mexico. Why do I say 32 to 35? Well, it all depends on what you call a species. So it's not even clear what a species is sometimes, but roughly 32 species. The closest relatives are in southwest USA, so almost certainly they're of North American origin, and they've moved down, the ancestors moved down into central Mexico, where they diversified into about 30 species or so. To answer this question, we've got to bring together information on the biology of the species with information on the Earth, history of the Earth. And just to give you a sort of conceptual model of how we go about this in the broadest sense, not a particular methodology, but a broad conceptual approach, is that we've got information on Earth history, volcanics, tectonics, patterns of erosion, climate, things that affect rivers and fish. We've got information on, I should also say, of course here I should say river channels and basins and things like that in the past, lakes. We've got information on the organisms that live across this landscape, where they're found, or how they're related to each other, what ecological preferences they have, and some, as I said, their sex lives, their behavior. So from the Earth history data, we can build space-time models of environmental change through time. From the organism data, we can build models of what we call a phylogeographic pattern, which is the distribution, the combination of the distribution where organisms are found and their evolutionary relationships. So the phylogeography is the combination of the phylogeny and the geography. Okay, we'll see that explicitly in due course. From those, we've built these space-time models of the environment and the evolution of the organisms. We then try and put those two models together to try and look for spatial and temporal congruence between pattern in both of these, in the environment and patterns in the evolution. And when we've got congruence and we can identify commonalities, then we can then try and infer either the environmental drivers, the things that drove the evolution, so which ones of these caused this, or which ones of this caused this, and the actual history of the organism in space and time. So those are slightly different things. One thing is what drove the diversification, and the other thing is what actually happened here, when, and where. So we start off, I have another 5 minutes. So we start off with, this is the kind of stuff that my colleagues, this is a paper published by, including Mike Ritchie and Constantino. This is the evolution, these are the fish, this is the evolutionary tree of the fish. The common ancestor is estimated at about 14.7 million years, which is in the late Miocene for anyone who's interested. And if we take this map, made by myself, I hasten to add, and we see these groups, so we can see D, this group up here, sorry, the square is a bit outlined, this group, this is called clade. A subtree is called a clade. Whenever I talk about a clade, it's a subtree. We can see D is found, it's the striped pattern, D is found in this distribution. We can see G, wherever it is, or H. So H is this shaded area, and H is this group of species from this node all the way down. Okay, so there is some correlation between, or quite a lot of correlation between the groups of species, the clades as we call them, and the geographical distribution. So that's kind of where we started. Tino and his colleagues spend a lot of time out catching fish across central Mexico. This is just a map showing the number of species that have been caught at these various locations. We can see that the highest diversity is found. This is elevation, by the way. This line of mountains here is called the Trans-Mexican Volcanic Belt. It's up to 15,000 feet high, highly volcanic area. We've got this is called the Mesa Central Plateau, this area up here, area of high plateau area. So here's the geography. That was the evolutionary tree. How do we put them together? That's the question. This is just give you a slightly better understanding of the geography of Mexico. So as I say, we've got the Trans-Mexican Volcanic Belt coming across here. We've got a large river system, the Rio Lerma, that drains most of the what's called the Mexican Plateau, high plateau that lies between two mountain ranges, the Sierra Madre Occidentale and Orientale. And it's this area that we're particularly interested in. So I'll just go through the, as I say, highly volcanic. This is Mont Colima in eruption. It's constantly in the news, Mont Colima. It's an area of great topological variation, huge great river gorges. We've got lake systems. Indeed, Mexico City used to be built on an ancient lake system. It's now all been covered up, Tinotajuan. We've got, this is Lake Lago Ziruan, and I don't know how easy it is to see, but can you see this swirl here, like that pattern there? This is Google Earth. Can you see those swirls? This is a giant lava flow that came down, blocked this valley. There used to be a river running through here, down through to the south. Then this huge eruption occurred, I think about, if I remember correctly, it's about 4,000, 5,000 years ago, so you can still see bare rock here on the imagery. This blocked this river channel and the lake formed behind it. So that's an example of the interaction, how the volcanics has driven the change in the hydrology of the system. And of course, the fish live in the rivers, so as soon as we've got this blockage, the fish here and the fish here can't mate with each other anymore, and they start to diversify. And that's how, to a certain extent, speciation in these fish occurs. I'll get to a nice spot of stopping. So we've got, central Mexico. We've got some information on the history of the river channels and paleo lakes in the system. So this is 15 million years ago. No, that's more like 20 million years ago, 15 million years ago, 3.5 to 5 million years ago, Pleistocene, 1 million, who knows, the present day. We've got information on the volcanics, and in particular a detailed digital volcanic map produced by Luca Ferrari, another geologist at UNAM. We've got information on the past where the channels used to flow. This data, for example, most of these were drawn by the ex-chief water engineer for Mexico City, whose hobby it was going out in the weekend and mapping paleo river channels across central Mexico. Very useful, a lot of information. We've also got things like cross sections of lake levels, geological sections of lakes and stuff like that, showing the depths of the lakes at various times and things like that. We've got a mishmash of information. We've got information on tectonic changes, fault lines, all this kind of stuff. Geography of speciation. Let's just do this with it and then we'll have our break. Okay, so how does species, where's the geography in speciation? Okay, so this is probably the most common way in which speciation occurs. You have an ancestor, wide-spread ancestor. Some new barrier forms between the population, maybe through continental drift. One side of the part of the population is on, say, North Africa, and the other one in Africa, and the other was South America. As the two continents split, they would diverge because there was no interbreed. Also, building mountains splits it. Volcanic eruptions, as we saw, could split a population in a river. Climate change can split populations, no problem. Long distance dispersal. In this case, the barrier's been there all along. The ancestor was only on one side. Perhaps there's a particularly strong wind. It blows a couple of individuals over the mountain range. They can set up a new population, but because it's a very, very rare event, then they start to differentiate and we end up with two species, one each side of the mountain. So that's another way speciation can work with geography, with other geography. Rivers. Rivers have a complicated system. So we're talking about fish that can't move between river basins. Okay, they're basically stuck in their river basins. They're not able to walk over to the next door basin. Now there's a process called geomorphological process called river capture, which occurs in river basins, and it basically works like this. So here's the original situation. This is a real example in North America, although the species on the right-hand side is just hypothesis. We've got the Heatherdale Creek here, flowing this direction, and then we've got the Mullum Creek here. And as you can see, this is an eroding creek. it's eroding its way this way, and eventually it cuts into this stream, and because it's got a steeper gradient than this stream, the water which used to flow down there now goes down there. And all the fish that were up here, upstream of the join, can now swim down into the Mullum Mullum Creek, and all the species in Mullum Mullum can go up into the bit of Heatherdale that is now Mullum Mullum. Nobody in the lower part of Heatherdale can now get back into those two areas. So what that means for a species, shown in this diagram, so if we have species H in Heatherdale, part of Heatherdale, the top bit of Heatherdale, has been grabbed by Mullum Mullum. All the species in there, individuals in there, are now in this one. They cannot mate with the ones left in the lower part of the stream. They differentiate, and through time we end up with a new species. Whereas the species in Mullum Mullum can never get across this way. So it's an asymmetrical change. So if you're in Heatherdale, you can spicariate, you can end up with two species. But if you're in the recipient basin, then you can't. All you're going to do is disperse potentially into a new area. Those are the geographical modes of speciation, or the ones that we're mostly interested in. The problem we have is if we're trying to infer what's happened in the past from what we see to present day, is that the same tree, pattern of tree in geography, so here's the species A, B, and C. So we're inferring, so this is what we see, okay, in geography, in the various basins. This is the evolutionary tree. And if we go back in time, there are two different histories that can result in the same pattern that we see today, both tree and geography. So here we can have a vicariant model. Maybe there was the ancestor E was found everywhere, then A broke off, split from D, A split from D, and then later D split into B and C, B and C. So we end up with A, B, C. But equally possible, maybe E was just in some ancestral basin here. It then managed to get into this basin, then managed to get through into this basin through river capture events, and we end up with exactly the same pattern. So if we see this, how do we know that the history is this one or this one? That's a problem. Okay, there are some other ways of speciation. There's one called parapatry, which is basically you get an environmental gradient across a population, and then there's environmental selection either side. Sexual selection, going back to good old sex, what you can get is if you get basically if you get differential mating between groups of individuals in a lake, you can get huge radiations. I don't know whether any of you know of the lake cichlids in Malawi and various African lakes. You get thousands of species that have all evolved in the same lake within very short periods of time, and it's all to do with sexual preferences of the various fish. This is a good place to stop after this one. So this is the kind of, again, a schematic diagram to show the sort of situation. Okay, here's our evolutionary trees, here's geography squished down to one dimension. So these are the kinds of questions that we need to ask to answer the big question. Is we're looking for differences within a tree as to big divisions. So we find one group of species over one side of the mountain, one group of species over the other side. So we're looking for that sort of patterns. We're looking for multiple trees of independent species showing the same type of pattern. We're looking for congruence between these major divisions. We're looking for things like that. Okay, that's the kind of patterns. Right, let's have stop there. So bypassed, unless you wanted to. I can continue quite happily, but if you want 10 minutes. 10 minutes? 10 minutes, okay.